Hello, everyone, and welcome to the podcast, Life is a Story We Tell Ourselves. I'm your host, Don Murphy. Joining us on this episode is Dr. Dale Greenwalt to discuss his new book, Remnants of Ancient Life, The New Science of Old Fossils. He will also talk about the importance of biomolecules as a means for exploring the ancient past. Dale received his PhD in comparative biochemistry from Iowa State University and worked in the pharmaceutical industry for many years. However, the rest and best of the story comes after Dale retired and began volunteering for the National Museum of Natural History at the Smithsonian in Washington, D.C. There he worked for the past 15 years curating the fossil insect collections. He initiated the Kishinin Formation Fossil Insect Collection Project and has been responsible for all field work and curation of the collection and interaction with and reports to the U.S. Forest Service. Dr. Greenwalt has a long list of publications and patents. Dale, welcome to the program.
Thank you for inviting me, Don. It's a pleasure.
Yeah. So, Dale, was there a particular occurrence or experience or epiphany that led to you taking the life's journey that you're on now?
As I think back on it, yes, there were a couple of them. One a long time ago and one fairly recently. Perhaps the first was when I was very young, in my early teens, when I visited my father's workplace. And my father grew up in the Depression, and when he got a job, he was deathly afraid that he might lose it. And so he kept this one miserable job that he had for his entire life, working in a smoky, very unhealthy foundry environment. And the one and only time I visited his workplace, he told me that he never wanted me to work in such a place. And at that, I decided at that point that I wanted eventually to get a job that was both one that allowed me to work in a healthy environment and one that would be intellectually stimulating. And so from that point on, I geared my education to going to college and getting an advanced degree. And I thank my father for making that comment some 60 years ago. It certainly has dictated the rest of my life. And more recently, and something that is more related to what we're talking about today, when I first saw the fossil of a blood-engorged mosquito, it was one of the most interesting, amazing moments in my life, because I realized that this was something that no one on the planet had ever seen before. I was the first one to see a fossil of a blood-engorged mosquito. And as I did research on that specimen, the last 10, 12 years of my life has been pretty much dictated by the fact that I found that particular specimen. That includes the publication of Remnants of Life. Those two events were very, very important in my life.
So what was so fascinating about finding a blood-engorged mosquito? Why did it affect you so? And it certainly harks back to, and people are fairly familiar with what you're talking about, because it was central to the story that Michael Crichton told in Jurassic Park. What went through your mind? I mean, what was so fascinating about that?
There were many things. The first was that I, in collecting specimens in the Kishinin Formation, which is in northwestern Montana, for 15 years, I knew that there were fossil mosquitoes there. And in fact, it's one of the best places in the world to collect fossil mosquitoes. But none of them were blood and gourds. And we've all swatted, mosquitoes in our life, and we realize how easily they can be squished when we hit them with our hand. And one of my thoughts was, how could this possibly, a blood and gourds mosquito with an abdomen blown up like a balloon, how could it possibly be preserved in rock as an intact specimen? And I wanted to find out how it was preserved. I also knew that this was the first blood engorged mosquito fossil that had ever been found, and that because of Jurassic Park, just as you said, Don, it would get a lot of publicity. People would be asking, does it contain dino DNA? Which of course it didn't, because it was 20 million years younger than the extinction of the dinosaurs. And so for a number of reasons, it was very, very exciting.
I understand. Well, so the other question that comes up every time we're talking about fossils or ancient biomolecules is exactly how did you find out how old this mosquito was? You were talking about it's 20 million years younger. That sounds awfully exact. What do you do to find out how old these fossils actually are?
When I retired 15 years ago from 30 years of being a biochemist, and started to volunteer at the Natural History Museum, I was very aware of what I didn't know, and that's one of the things I keep telling myself is I'm proud of the fact that I know what I don't know, and I'm willing to admit that. I am not a geologist. And the dating of the Kishinean Formation was done certainly not by me, but by geologists who have been researching that area for the last half a century. And they simply looked at the various layers of rock, and knowing that certain species, particularly the mammals that live there, occurred during very finite periods of time, they were able to determine that these rocks were from what we call the Middle Eocene, which is around 50 to, say, 40 million years ago. In addition, there are ways that a geologist can take minerals from the rock and do chemical analyses on them and determine what age they are. So this work was done by other people, by geologists who know what they're doing, and the Kishinean Formation. The oldest portions of it are about 46 million years old, but the lake sediments accumulated for some 10 million years thereafter, and the youngest portions of it are maybe 35, 37 million years old.
Now tell us where this formation's located.
I think the best way to answer that question is to talk about Glacier National Park. Most folks who hear the podcast will know where Glacier National Park is. It borders Canada, of course, up in the northwest portion of Montana. But the river, the Flathead River, forms the western boundary of Glacier National Park, from Canada all the way down to the Bob Marshall Wilderness. And it is the Flathead River which is eroding its way through the sediments of this ancient lake, and in some places exposing rock that contains fossils of organisms that lived back during the Middle Eocene. And it's interesting in that there are some types of rocks that will preserve mammal bones, some types of rocks that will preserve, for example, shells like clam or snail shells, and some types of rocks that will preserve insects. And the formation is, I think, at this point and in the future will be most famous for its beautifully, exceptionally well-preserved insects.
Well, you talked about preservation. It's an interesting segue to mention, as an aside, how important it is to preserve state and national parks, and in this case, national forests, because they're tremendous repositories of information, scientific information. I know your book talks about information that was found out at Yellowstone National Park, and certainly at other national parks around the country and around the world for that matter. They're great repositories of scientific information, and preserving them for future generations is not only important for recreation and enjoyment of their scenic beauty, but for the scientific information that they will yield as well. So really appreciate the work that you were able to do there. Blood-engorged mosquitoes, talking about blood, that must have led you to this fascination that you have with biomolecules. So how did this blood that was preserved for millions of years inside this ancient mosquito lead you to such an interest in biomolecules?
It's as you say, Don, an interesting story. The paper that was published on the blood-engorged mosquito, which presented data that definitively identified certain parts of the hemoglobin molecule still within the abdomen of the mosquito. When that paper was published, there was quite a bit of publicity. And about a year after that, I was contacted by a couple of young academics who wanted to publish a 2-volume book about the fossil record of parasites and parasitism. Believe it or not, you wouldn't think that there would be a fossil record of parasitism, but it's actually quite good. They wanted me to write a chapter on fossil blood. And so I did that. And during my research in preparation for the writing of that chapter, I delved into the vast amount of work that has been done in the recent past by people who have identified not just ancient blood molecules, but ancient biomolecules other than blood. And let me define here: a biomolecule is simply a molecule that is unique to living organisms and is required for the function of those living organisms. And as you mentioned earlier, Don, examples are DNA, proteins, pigments, structural molecules like cellulose. And after that chapter was published, I began to think that, wow, there might be enough material here for a book. But even more importantly was that I was absolutely fascinated by the preservation of biomolecules in these rocks. I mean, rocks are supposed to be, fossils are supposed to be, totally mineralized, and they're made of rocks, and the biomolecules were supposed to have been degraded, millions and millions of years ago. And so I convinced myself that if I was interested in this, if I found this fascinating, well, then the lay public would also find it fascinating. And so that led to the writing of Remnants of Ancient Life, which is directed at the lay public. It's not meant to be a technical academic book. I really wanted to let the lay public know about all these fascinating biomolecules that were out there.
All right. Well, let's talk about one biomolecule, and in particular, you already mentioned hemoglobin, but let's focus on heme, which, of course, is a subset of the entire molecule of hemoglobin. And what you found out, what you were able to extract, the information, the biological information you were able to extract from the blood that remained in that mosquito for millions of years. I mean, it's just totally mind-blowing. So could you maybe let our listeners know exactly what you found out with respect to the biomolecules that remained in that blood?
Of course. So the hemoglobin molecule is responsible for carrying oxygen in our blood, in our circulatory system. And most of hemoglobin is a protein. And it depends, however, on other non-proteinaceous molecules to actually carry the oxygen. And those molecules, which are not proteins, carry oxygen via their binding to iron. And so you have the large hemoglobin protein made of protein, and within each hemoglobin molecule, there are 4 heme moieties, and each heme moiety has 1 atom of iron. And so I knew that perhaps the first thing we should analyze for would be the iron, because that would be easy to find. And if there was an abnormally high level of iron, that would suggest that a larger portion of the hemoglobin molecule was still there. And so that was relatively easy to do with the help of the Mineral Sciences Department at the Natural History Museum. We found immediately that the abdomen of the blood and gorge mosquito contained really, really high levels of iron. And we had a beautiful control, because we had other mosquitoes which were not blood engorged, and they did not have these high levels of iron. And in fact, even other portions of the mosquito that was blood engorged, for example, the head or the wings, were very low in iron. So the high iron content was very specific to this dark material that was in the abdomen of the blood engorged mosquito. We did not find any hints of the protein being preserved, but the very fact that we could prove definitively that the heme molecule with its iron was still there was enough to prove that these were, in fact, blood engorged mosquitoes.
That's just absolutely fascinating that you were able to extract that kind of information from the mosquito. And for our listeners, just for a little bit of context, if you see rust anywhere on iron, you know that it has a dark reddish color, and that's an oxide of iron, or iron oxide. And it's actually the same thing that's inside the blood engorged mosquito. Iron binds oxygen readily, and that's why you get rust on iron. And so that'll give you a little bit of context of what we're talking about here. It's just absolutely fascinating.
And the miracle of hemoglobin is that it can bind iron and then it can release it. And so in our circulatory system, it binds iron in the lungs and releases the oxygen at various parts of our body as needed. And there's an interesting story about that in the book, about research that was done by people looking at the hemoglobin of mammoths and how tightly or loosely it bound oxygen relative to today's, for example, African elephants.
Right, and you can get build a relationship, and that's a good segue to talk about why these biomolecules are important. So, when we talk about dinosaurs and we talk about modern animals, and we talk about taxonomy and phylogeny, we're really talking about morphology and structure. And when we try to piece together the connections that go from now and animals that exist now all the way back to their ancestry millions of years ago. But now what's important is you have these biomolecules that you can use instead of morphology or the structure of the bones and that sort of thing, which you usually look at. That's just revolutionary, isn't it? I mean, this is new science.
Oh, absolutely. The morphology has carried the science of paleobiology a long, long way for the last couple of hundred years. But now there's this realization that within what we used to think was 100% rock, there are biomolecules that we can use to tell us a number of things about the ancient organisms that are now long extinct. And some of these biomolecules can tell us what type of behavior these organisms had. There is one particular protein that is sexually dimorphic, and if the only specimen you had was a tiny little piece of bone, if you could extract that protein, you could determine if that piece of bone came from a male or female organism, something that you could never do based just on the structure of that specimen. So
when you say sexually dimorphic, you mean that some of these biomolecules are sex-linked. In other words, they're on the sex chromosome of a particular animal, and if you can extract that particular molecule, you will know whether it came from a male or a female. Is that what you're saying?
Actually, in this case, Don, the female protein has a slightly different sequence than the male protein.
Mm-hmm.
And so when you sequence the protein, the sequence itself, the order of the subunits of the protein that go into this long little beads on a string structure that proteins have, this sequence of those subunits is specific to the sex of the organism.
Wow. So that's absolutely fascinating. But you just mentioned, something I think that's also mind-blowing, that you can use these biomolecules to determine the behavior of a particular species long ago. Tell us how that works.
Well, there's a number of examples. And for example, there is a 340 million year old crinoid. Crinoids are marine organisms, lived in the ocean, and they are very closely related to today's starfish, and feather stars, the echinoderms. And a pigment has been isolated from fossil crinoids that are hundreds of millions of years old. That pigment is bright red. And what it tells us is that these organisms, what color these organisms were, and that they lived in an environment where they were probably using the pigment to warn potential predators away, because the pigment not only is red, but it's toxic. And so the organism living at the bottom of the ocean was not mobile. It was attached very firmly to the bottom of the sea, and so it was very susceptible to predation. But it had evolved this toxic pigment that, again, was not only toxic, but it was bright red to warn away predators. And so that's one example of how a pigment can tell us, in this case, that the crinoids use these pigments to warn away potential predators. Another example, and this is, I like this particular example because it's really very simple, and it involves a bacterium, Yersinia pestis, which is the cause of the Black Death or the Black Plague that was so terrible back in the early Middle Ages. The bacterium has been found in human bones that are 500 years old, 1,000 years old, 2,000, 3,000, 4,000, 5,000 years old. And the sequence of the DNA in that bacterium demonstrated that about 3,000 years ago, about 1,000 BC, there was a single mutation. And that prior to that mutation, the bacterium really did not cause a serious disease. But around 1,000 years ago, there was this one single mutation in the DNA of this bacterium that allowed it to dissolve blood clots in people who were infected with the bug. And as a result, it was able to circulate throughout the entire body of its host, the person infected. And so it became much more virulent and led to what we now know as the Black Plague. And so this is an example of a single mutation that was responsible for the evolution of this bacterium from a relatively innocuous organism to one that caused literally tens of millions, if not hundreds of millions of deaths during the Middle Ages.
And so while that was extremely deleterious and harmful to human beings, that single mutation in the bacterium afforded that bacterium a tremendous advantage for survival and reproduction. And that can be scaled up. That happens even on a grand scale with something as large as a mammoth that might have to adapt to the cold. A single mutation in one gene can perhaps cause more hair to grow on the foot pads of a particular species like a mammoth and give it an advantage in the cold. And then that advantage is passed on to future generations of mammoths, and their survivability is increased. And it's just a wonderful example of how not only evolution takes place, but also the etiology of diseases and how diseases can spread from a single mutation that gives a bacterium a particular advantage. And that just teaches us so much more about how even diseases advance evolutionarily.
Absolutely. And the example of the mammoth is one of my favorites, because in that situation, scientists were able to recover genetic material from a mammoth that had been dug out of permafrost in Siberia. And once they had the DNA sequence of the mammoth's hemoglobin, they were actually able to use laboratory techniques to synthesize the actual protein. So what they had in their test tubes, so to speak, in their laboratory was mammoth hemoglobin. And once they made mammoth hemoglobin, they were able to test it to see how it compared to, for example, African elephant hemoglobin. And what they were able to show was that a mutation made it much easier for the mammoth to release oxygen in the peripheral circulation of the mammoth. And so, living in an ice age, it's very important to keep your feet warm. And that was the exact function of this particular mutation.
Mm-hmm. Fascinating. Well, there are so many biomolecules to talk about that. And in your books, I really encourage people to pick up a copy of your book. But one particular protein that for me was fascinating that you talked about was opsin, which has to do with color vision. And you talk about that quite a bit in your book, and the perception of color and how that evolved over eons. And I have some. It leads to some philosophical questions too, as to whether or not animals perceive color or react to color the same as human beings do, or get the same feeling. So let me put a finer point on the question, because it's a little weird question that popped into my mind. So there's this philosophical concept that's known as qualia, and philosophers use it to describe the nature or content of our subjective experiences. So my question has to do with, there's this biomolecule opsin that you talk about in your book that gives color vision, and then paleobiologists draw certain conclusions about what the perception of color meant to the animals. But then my question is, how much are we ascribing human reactions to color to these animals, or is there some anthropomorphizing going on when we try to understand how ancient species might have used color vision? I know it's kind of a convoluted, complicated question, but it just occurred to me that has to be something you would consider as a paleobiologist. Is that so?
Yeah, that whole area, Don, is I'm glad you brought this up, because it's really fascinating. We have to ask ourselves the question, if in fact organisms could not see colors, there really would be no need for colors. And in fact, the evolution of color vision occurred really, really early in the evolution of complex organisms. Well over 500, 600 million years ago, organisms had color vision. And the example of the red crinoid. I don't think there's any chance here that we could be accused of being anthropomorphic when we talk about a crinoid advertising its toxicity to potential predators. So, yeah, the presence of pigments combined with the ability of organisms to see those pigments creates, an infinite number of interesting stories. Camouflage, is very important to, and even to the dinosaurs. And there is a lot of evidence now that many of the dinosaurs, which were subject to predation, were camouflage, and they used different colors to provide that camouflage. And obviously, that tells us something again about the behavior of the organisms. If something is well camouflaged, it, for example, may in fact be pretty slow. It didn't have to run very fast. It relied on its camouflage to escape predators. You can go in all kinds of different directions when we start talking about colors and pigments in the fossil record. It's fascinating.
Yeah, absolutely. Well, that leads to the particular pigment that is well known, at least amongst human beings, and that's melanin. And you talk in your book a great deal about the evolution, the development of that particular biomolecule, that particular protein. It's a pigment. Can you talk about the role of melanin? It played, it seemed, a huge role in the evolution and the development of all life on Earth.
It's a very important compound all across the diversity of life. The first fossil record of melanin itself is something like over 300 million years old. Melanin was isolated and proven to exist in the ink sac of a fossil squid. Again, a situation where that tells us that the squid was using this black pigment, melanin, as a defense. It would spray the black pigment out, just as living cephalopods do today, to create a black cloud that would allow them to escape from predators. And the fossil record of melanin obviously is present in large numbers of organisms. It has not been isolated from very many dinosaurs, however, and we know now that dinosaurs had feathers. We know that those feathers were colored, and they contain melanin, but it was not because we were able to isolate the actual biochemical melanin. Rather, we were able to visualize the little packet-like structures that stored the melanin. And there's a lot of research which indicates the different size of these little storage packets can be related to the type of melanin stored, and different types of melanin, of course, dictate different colors. So, yeah, everything that we all one thing that we all want to know here is what colors were the dinosaurs. And there's a lot of information that some of them were very, very brightly colored with bright red and other types of colors.
But you haven't recovered any actual dinosaur pigments, is that right?
Not from the dinosaurs, but we have been able to recover it from other organisms.
Understand. So the other biomolecule that's extremely important that you talk about in the book is collagen, and most of our listeners will know they see collagen advertised on television as almost a cure-all for every joint in your body. So people know collagen is important in that respect, but as a biomolecule and telling us about what happened in ancient times and how organisms may have behaved, collagen, there's something called collagen fingerprinting. Can you tell us about that, how that developed? Because that's a fascinating story, and it actually is a powerful tool now that's also used in trying to determine and understand how life evolved and the structure of particular species in ancient times. How does this collagen fingerprinting work?
It's a fascinating technique that was developed by a laboratory in England, and an individual by the name of Mike Buckley and his colleagues. Collagen, of course, there are a dozen different types of collagen, and they comprise most of the mass of things like ligaments and tendons, and they're essential for life. They are essentially responsible for connecting the bones to muscles. And of course, if that connection didn't exist, we would simply be a flat, round blob on the surface of the ground. So what Mike did was he developed a technique where he could take a bone and extract from that bone a particular type of collagen. Now, these bones, the oldest of these bones is about a million years old or so, but that gives us a lot of time, particularly important time, because it is within that time period that our own species evolved. But what Mike did was develop a technique whereby he could take that collagen. He didn't have to sequence it, but he could just fragment it and separate the different fragments of collagen based on size. And that pattern of size fragments would make a fingerprint that was specific to different organisms. So the array of collagen fragments from a pig would look different from the array of collagen fragments from a deer, for example. Now, this is particularly important when you are an anthropologist or a paleontologist and you're digging in the floor of a cave where 100,000 years ago humans lived, or Neanderthals lived, and you harvest, you dig up literally hundreds and hundreds, maybe thousands of different tiny bone fragments. And these bone fragments really don't have very much morphological information to provide. And so it's very frustrating because you have all these little bone fragments, but you don't know where they came from. But what you can do is very quickly analyze each of these tiny fragments for a collagen fingerprint, and you can tell very quickly what organism these bone fragments belong to. And so you can, it tells us a lot about the diet of the Neanderthals, what organisms they were hunting and consuming. And it is a technique now that is used universally by anthropologists and paleobiologists throughout the world, because the world's museums have literally tens of thousands of drawers that are filled with tiny bone fragments, and no one knows what organism they came from. And here's a technique where we can very quickly and very definitively identify the organism from which these bone fragments came from.
Yes. That's just absolutely fascinating, this story. So, Dale, maybe you can share with our listeners what you think this revolution is going to lead to. I mean, now we have this tremendous technique of biomolecules and analysis of biomolecules, and they're telling a story. What new have we learned so far, and what have we yet to learn that you think these biomolecules will reveal to us?
One of the more interesting things about reading the literature is papers which extend the age of specimens wherein we can find biomolecules. Biomolecules are usually very, very fragile. They degrade very quickly, and different types of biomolecules are more fragile than others. So, for example, the oldest DNA is less than 2 million years old. The oldest protein is just slightly less than 4 million years old. But when we get into less complex biomolecules like melanin or the red pigment of the crinoid, now we're talking about small biomolecules that can exist for literally hundreds of millions of years. So the younger the specimens are, the more likely we are able to extract DNA and proteins, which are, of course, genetic material, and they can tell us so much about an organism. Once we can sequence, and in many cases we have, in fact, sequenced the entire genome of a fossil organism, that is, every DNA molecule that ever existed in that organism, it's Katie bar the door in terms of what we can find out about that organism. So it's kind of a race, where we're trying to find DNA and proteins in older and older rocks, in older and older specimens. And it's interesting to see reports of the ages as they are extended deep into time. I believe very firmly that we will eventually be able to extract DNA proteins from far older animals. And this is going to provide us with a huge wealth of information about extinct organisms that lived before, long before primates even evolved. But to me, the most exciting part of ancient biomolecules is not in the preservation of the actual biomolecules, but something called ancestral sequence reconstruction, where we can study how mutations occur and what patterns they occur in, what residues are most likely to be mutated. And using computer algorithms, we can predict the sequence of ancient proteins in the absence of any fossil remains. And the scientists who have developed and who are improving these techniques are opening up a fascinating area whereby we can predict what proteins were like literally hundreds of millions of years ago. A good example is, a lot of people believe that life evolved near or in hydrothermal vents in mid-oceanic ridges. I think most of us have heard of the black smokers, these vents where in the middle of the ocean, which are venting steam from the interior of the earth. And people believe, well, that's a very rich environment with a lot of nutrients and whatnot, but it was very, very hot. And if, in fact, life had evolved in those vents, those organisms must have been able to tolerate really high temperatures. And what scientists have done is they've taken proteins from living organisms that work best at, our room temperature, and they have predicted what the sequence of the protein would be in organisms that would have lived in these hydrothermal vents. And when they put these proteins together and they sequence them and they synthesize them and they test them in the lab, lo and behold, it shows that these ancient organisms or these ancient proteins that have been predicted by computer algorithms do, in fact, function very nicely in boiling water. And so these techniques can open up vast amounts of information in the total absence of the fossils themselves.
Well, that is so mind-blowing, because you think about time machines and we're always talking about, traveling into the future, traveling into the past. And so now, instead of predicting the future, we can use these techniques to predict the past. I mean, it's hard to wrap your mind around that. That's just mind-blowing.
Yeah, I can't wait to see what new information will be gleaned from the fossil record and from the ancestral sequence reconstruction in the future, in the near future. I mean, this new science is really growing exponentially, and interestingly, it is attracting young scientists to the field. And I think that is just really, really cool that the younger scientists are realizing the potential of ancient biomolecules to tell them things in addition to what simple morphology can. So, yeah, the future is going to be very, very exciting for paleobiologists.
All right. Well, we could talk about this all day, and there's so many more. There's so many more biomolecules to talk about. But this being Life is a Story We Tell Ourselves podcast, I always try to wrap up by asking kind of a transcendental question, and so in all of this journey that you've taken into paleobiology and into biomolecules, has there been some event or something that's happened to you that you would characterize as transcendent and that changed you in some way that you never imagined you'd be changed as a human being, whether it be spiritually, physically, or just psychologically?
Boy, that's a tough question. I have been exceedingly lucky, and I am very thankful that I was given the opportunity to work as a volunteer in the Paleobiology department of the Natural History Museum in Washington, D.C. I started out as a volunteer, but I was able eventually to talk my way into an independent project, which in fact was the collection of the insects from northwestern Montana. And I've been doing that now for 15 years. And I will certainly, I have every intention of continuing to do this as long as I live. And the reason is, there are many different reasons, but one of the most interesting reasons is that when I'm out collecting, and you can imagine yourself sitting near the river and you get a piece of rock and you take a rock hammer and you split it. At that very moment, when you split that rock, I am revealing something that has the potential of being totally new, unknown to the human race. No one else in the world has seen what I am about to see when I split that rock and see what's there. And that is the thing that keeps me coming back. And, because it could be something as interesting as an organism that contains biomolecules and opens so many different avenues to our ability to understand early life on this planet. It is something that is very unique, and I think something that perhaps all of us aspire to see something that no one else in the world has ever seen, to do something that no one else in the world has ever done.
Well, Dale, this has been a fascinating conversation, and not only that, it's been inspiring. I mean, to think that you've done all of this in retirement. So, all of you retirees who are out there listening, life is just beginning at retirement. I think that's the lesson that we can take from this. And there's so much you can accomplish in your life after you've retired, and so many contributions that you can make. And Dale, we certainly appreciate the contributions that you've made in the tremendous discovery and directing paleobiology into an entirely new and revolutionary direction. Dale, thank you for joining us. And I'm sure there's another book in you that will be coming out soon. Thank you, Dale.
You're very welcome, Don. I appreciate the opportunity.
Thank you for joining this episode of Life is a Story We Tell Ourselves. You've been listening to the scientist and author Dale Greenwalt discuss his new book, Remnants of Ancient Life. If you would like to learn more, you can subscribe to the podcast at lifeisastorypodcast.com. Don't forget to hit that like button and leave comments or questions for Dr. Greenwalt. Be well, share happiness, and remember, never stop questioning. Curiosity has its own reason for existing.